Hello, good afternoon. We will be finishing up the note today on DNA replication, and that's actually going to take like a good half of today's class, and then we will start on the second half of the lecture, which is the cell cycle and mitosis. Judging from my morning class today, we're not going to be able to finish lecture six, and that's okay. Some lectures takes longer than others. So whatever we don't finish today, we're gonna finish it next week. Okay, so let's do a quick recap as to where the story is so far. We're talking about DNA replication, which is copying all the genetic materials before a cell divides. And we are specifically looking at how this happens in prokaryotes, right? Because it's easier to study them than to study animal cells. And then when we finish talking about this, we will compare the differences between what happens in the bacteria versus what happens in us. So we know that DNA replication follows the semi-conservative model, which is you would separate the parent strand and then you would use each half as a template to create the missing half of the DNA. Yeah, so in the end, you will have two copies of the DNA. Each copy will have half being the original material and half being the new material. So to do this, you will need various enzymes to help with the process, starting with this thing called a helicase, which opens up the bubble, creates the replication bubble, the origin of replication, and uh, and then it will continue to move sideways to make the bubble bigger and bigger and bigger. But as you're doing this, as you are unzipping the DNA, you actually increase the tension in the double helix, and we need gyrase, the green enzyme here, to reduce that tension such that the DNA is not gonna snap and break, basically. Okay, and so those are the two enzymes that are going to kind of initiate the process. But once you've separated the DNA strands, the two strands are attracted to each other because of the hydrogen bonding. The A is going to be attracted to the T's, and the C is going to be attracted to the G's, and the strand have a tendency to come back together. But we don't want them to come back together, right? We need to keep them separate so we can use the template to create the missing half. And that's where the single strand binding proteins are going to be important. It binds to the separated DNA strands and prevents them from coming back together, from closing. And then towards the end of last class, I introduced you to this enzyme called DNA polymerase 3. So DNA polymerase 3 is an enzyme that is needed for building the new DNA strand. And what it actually does is it will use the parent strand as a template to build the complementary DNA strand. Maybe I'm just going to add that extra bit um, for you to make it a little bit more clear. I'm just going to move this down. If you have room in your notes, okay, can say the DNA polymerase 3 uses the parent DNA as a template to build a complementary strand. The enzyme, however, has two limitations. Number one, it can only build DNA in the 5' to 3' direction, and it can only build on existing uh, DNA. It's not going to start on its own. Right? So one way to help you understand this is to give you an example. And like an analogy, okay? So like my kids, when they come home, they don't do their homework automatically, right? So like my younger one's like she's learning multiplication, right now. So for example, like she would have a work. You don't have to copy this down. I'm just trying to help you understand what's going on here. Okay, just gonna randomly make some question. Okay, so let's say she has a homework and she has to do all these questions. Okay, so she doesn't do it. Right? She's not gonna start doing the homework on her own. So what I have to do is I have to sit down with her and I'm like, okay, like what's the answer to this one? And then we do it together. It's like, oh, 21, and then this is going to be 16, and then this is gonna be 30. And then after I do a few with her, and then I'm gonna leave, and then she'll be able to do the rest herself. Okay? That's basically what DNA polymerase 3 is like. It's not going to be able to build on its own. It's not going to start on its own. It needs something to do a few nucleotides for it first, and just like I do a few questions for my daughter before they can take over. Does that make sense? Okay, so that's the limitation, right? All right. So in the context of what our picture here, something has to come along and do a few nucleotides. Okay, so that something is an enzyme. You can use whatever shape you want. This enzyme is called primase. Primase. It's going to come along and it's going to do a few base pairing, base pairs for DNA polymerase 3. It's like, okay, I see a C here, so C is gonna pair with G. Another C is gonna pair with G. A is gonna pair with T. Stay with me, guys. This is important. A normally pairs with T, right? But actually, the primase, instead of building DNA nucleotide, it's gonna be building RNA instead. Okay? Not sure why. That's just the way it works. And then T is gonna pair with A, and then A is gonna pair with U, and then it's gonna stop. It's gonna do a couple, 50, 60s of them. I'm not gonna do 50, 60 here, obviously. So it's gonna do some nucleotides, and then the DNA polymerase 3 will come along and basically continue the work. Is that okay? Do you understand what I mean by that? This is a key thing to understand before we finish the notes. Not really? Okay. Not really, as in, like, can you give me a question, Asterin? Like, do you have a specific question about why does it create the RNA? Okay. All right. So back to the situation here, right? The DNA polymerase 3 has two problems. Okay. The second problem is that it can only build on existing DNA. Right? Like my analogy, like my kids, they are not going to be doing their homework on their own. Someone has to sit down with them and do a few first, and then they would be able to continue the rest of the worksheet. Same thing here. DNA polymerase 3 is not going to start building the DNA on its own. It's not going to be like doing the questions on its own. The primase is going to come along and do a few nucleotides for it first, and then the DNA polymerase 3 will take over and build the rest of it. Okay, that's why we needed the primase to lay down a few nucleotides first. Anyone else have any questions? All right, this next part confused the heck out of my morning section, okay? So please let me know if you get it. If you don't get it, you need to ask because the rest of the lesson it kind of builds upon this next concept. So here we have some nucleotides. Right? Looking at the black strand, you see that the top strand is like 5 prime over here and 3 prime over here, right? So this strand here that we just built, if I need to give it some directionality to it, should I put a 5 prime here or should I put a 3 prime here? Which one would be correct? Is there only one correct answer? Is it 5 prime or is it 3 prime? I'm gonna do a poll to see what you guys think. You let me know. Okay. Is it A, 5 prime, or B, 3 prime? And if you're not sure, you can choose not sure. It's fine. Okay, so let me release the results for you so you guys can see. Well, it's quite a bit of people say it is going to be 5 prime, and that would be correct because DNA bonding, right? The property is that it is complementary, of course. A goes with T, C goes with G, but also they are anti-parallel. Exactly, Sheila. Anti-parallel, right? So if you see a 3 here, it has to be a 5 here, and it has to be a 3 here. Okay? The numbers are never going to be the same on both strands. If you see a 3 on one strand, the other strand has to be a 5, and vice versa. Are we good? Now, here comes the next weird concept people are having trouble this morning. The second limitations of DNA polymerase 3, and again, this is the enzyme that will actually build the majority of the complementary strand, right? It can only build DNA in the 5 prime to 3 prime direction. Okay? So don't draw this yet. Think about it. I'm going to ask you, when the DNA polymerase 3 continues the work, is it going to go this way to the left, I guess, towards the fork, or is it going to go that way, away from the fork? A or B? Let me do the poll again. Which way will we build the DNA in? Which direction? Okay, read the limitation. Okay, thank you. Um, so like I said, right, it's very a lot of confusion here. It says only build DNA in the 5 prime to 3 prime direction. So when we say build DNA, we're talking about the new strand that you're building, right? You see, this is this direction. This is 5 to 3, 5 to 3, right? So the DNA polymerase 3 is actually going to build like this, C, G, T now because we're building DNA. C, G, A, so on and so forth, right? So that's what it means by 5 to 3 on the new strand that you're creating, on the new strand that you are building. Okay, the other direction is actually 3 to 5. You see? 3 to 5. So that would be incorrect. Okay, I'm just gonna pause for a second, let you think about this, let it sink in, and ask for clarification if you need. Okay. You mean the direction? Is that what you want, Huyen? Okay, so do you understand why if this is 3 prime, this is going to be 5 prime? Is that clear, that part? Okay. So if you look at the numbers, do you, and like everyone else, when I say you, like, do you understand this here is 5 to 3, and this here is 3 to 5? I'm not looking at the original. I'm looking at the new one that we're building. This way to the left is 5 to 3. You literally just read the numbers. And then this way is 3 to 5, right? So DNA polymerase 3 can only build DNA from 5 to 3. Right? That's its limitation, right? And that's why it's gonna go towards the fork like this. It's only gonna be able to do it this way and not the other way around. Sheila, what do you mean by 3 to 5? Can you elaborate a little bit?
I'm just answering the question because since
Oh,
they
okay.
okay, okay. Sorry about
No, no,
that.
no worries. Thank you. So now let me try this analogy again here, okay? Let's say the worksheet is actually much longer, right? The worksheet is much longer. And then I'm just gonna throw in a few more questions, okay? The questions are not important, of course. Whatever, right? 1 times 9, 2 times 8, 5 times 7, okay? So I did a few questions with my kid, but she is only able to, for the sake of the analogy, she's only able to go down the list. Okay? She's only going to be able to go from top to bottom, top to bottom. So if I start the homework with her in the middle of the worksheet, she will only go top to bottom. She'll only go down. All these questions will remain undone. Okay? So how is it possible for her to do these questions? Well, if I want her to do these questions, I'm gonna have to sit down with her again, and I will do it with her again. Right? 5 times 7, 35, 2 times 8, 16. And then she'll be like, oh, okay, I get it. And then she will go from top to bottom again. That's basically what's
The
happening here.
analogy of the DNA, right?
I'm sorry, what?
Is that the basically the analogy of the DNA, how it
Yeah,
goes?
so like these are like the questions are like the parent DNA, right?
Right.
Parent DNA, and then like you're like building new ones, right?
Mm-hmm.
Okay. So take a look here. Take a look here on the other strand now. Okay.
Mm-hmm.
The primase is gonna come and it's gonna do a few nucleotides.
Mm-hmm.
A, U, A, right? C.
GG. Yeah.
Now, what number goes up here? Is it
Three?
five or I'm sorry?
Is it three?
Is it
Wait, if
anyone
it's
else? Or like you can change your answer to, I guess. Um,
Five. Five.
is it
yes,
five?
okay. This is five. Why? Well, let's take a look. If we go all the way to the left side, you see that the bottom one is 3, right? The black
Right.
one? 3 all the way 3. So if this is 3 at the black, this must be a 5.
Yeah, five. Okay, I get it. It
Okay,
has to be the
and
opposite. One, three, and
that's
one,
right.
three.
Exactly. So this is going to be 3 now, right? And
Right.
so that's the a few examples, so to speak. And now the DNA polymerase is going to come and continue to build. But my
Mm-hmm.
question is like, which way is it going to go? Is it going to go to the right or is it going to go to the left?
For
Is it going
that one, I think it's gonna go to the right.
What is
right?
left? Right? What is left?
Oh, is this gonna go to the right or is it gonna go to the left? Let's do another poll and see, okay? I'm
Mm-hmm.
talking about the bottom strand here, guys.
Mm-hmm.
All right, let me relaunch the poll. Just want to see what everyone thinks. When it continues, when the DNA polymerase 3 comes and continues to build the strand, due to its limitation, which way is it gonna go? Okay. It's better, the result, compared to last time. But a few people still seems to be struggling. Remember, the DNA polymerase 3 can only build from 5 to 3 on the new strand. This way, this way is 5 to 3. You literally read the number 5 to 3. So when the polymerase come, it is going to go this way. C, G, G, C, and then it's going to continue to go that way. Okay, remember, it can only build from 5 to 3 on the new one. Don't look at the black one. The black one is just a template. Okay, it's the new one that we're building. The new one has to be from 5 to 3. So in this case, it's going to be going away from the fork. No, this is not conservative. This is semi-conservative because you separate the strand, you use the black ones, the original, as a template, and then you're building new strand. All right. the numbers, the direction, like if you're confused, let me know what you're confused about so I can help you, right? Like I said, it's crucial to understand what goes where for the rest of the lesson to make sense. That's why I'm taking some time to make sure that you're not confused. Okay, so let's carry on. What about these nucleotides? How are we going to copy these nucleotides if the polymerase 3 cannot go backwards, right? Okay. So what needs to happen is we need to have another primase that will come and it will do a few more nucleotides. And again, this is also 5 to 3. And then when the DNA polymerase 3 comes, it will build again in the right direction. And when you open up a few more, as the helicase open up the DNA and expose more nucleotides, then you will put another one, and then the polymerase 3 can continue. Okay. So this thing right here, the examples, that is called an RNA primer. Okay. So basically, primase creates an RNA primer such that DNA polymerase 3 can build upon it. Okay. So that's an RNA primer, that's an RNA primer, that's an RNA primer. Okay. And again, the reason why we need to have multiple primers at the bottom is because of the first limitation here. The DNA polymerase 3 cannot go backwards. It cannot go from 3 to 5. It can only go forward. Okay, so as you unzip the DNA and expose more black DNA, so to speak, right? Like the black nucleotides, the parental nucleotide, you got to put more primers so that you can continue to replicate those things. Okay, that was a lot. Um, Anything at all you want to ask? It's okay, like that, to ask. Like seriously, like if you don't get it, you don't get it. So let me know what you don't get, and I'll tell you. Does the analogy help here, right? Like if you think about it, this is basically 5 prime to 3 prime. Can only go downwards. Right? So like if this is a really long piece of paper, and then like when you unroll the paper more, and then there are more questions, right? The only way to do more questions would be if I do a few more with with her, and then she'll be able to continue down. So as you unroll the paper, more questions get exposed, and then you will have to repeat this process over and over again, right? It's okay if we unroll the paper at the bottom, right? You can just continue to go down, right? So for the top strand here, as you unzip more, is no problem. I don't need more primer because this is gonna just continue to go on in the 5 to 3 direction. It's the bottom one that's causing a bit of a problem. I need to have multiple primers. Okay. All right. So these fragments that we're creating, and these are called Okazaki fragments. It's named after the person who discovers them. Okazaki fragments. This is like a super busy diagram, but I'm just gonna add one more thing to it. It's up to you if you want to add this or not. Okay, so this is just another enzyme, and this is the DNA polymerase 3. Okay, so like you would have polymerase 3 here, polymerase 3 here, polymerase 3 here. It's basically going to continue building upon the RNA prime. Now, in case you're wondering, like, who cares about these things, right? Like, why do we spend so much time learning about it? Um, during COVID, you probably remember there's this thing called PCR testing, right? You do a nose swab and then you send it to PCR testing to see you're positive or not, right? The whole process of how PCR works is exactly same as what we're learning here right now. Okay. So we're going to come back in lecture 9 to learn about PCR, and you will hear a lot of familiar terms when we get there that is similar to what we are learning right now in this lesson. So there's some relevance. All right. So let's start a brand new diagram to see how this actually works in a like a more simplistic way. Okay, so summary, I guess. Summary. So I need you to have a bubble because so far we have just been looking at the fork, right? Like one side. So let's draw a bubble. And 5 to 3 here. I just choose to put 5 to 3 on top. It could have been the other way around. It doesn't really matter. However, if I put 5 here, I hope you understand the bottom one has to be 3 to 5, right? So let's think this through again. Okay. Please use color so that your note makes sense. Because DNA polymerase 3 cannot start the process on its own, it needs a few nucleotide first, right? It needs a few examples, right? I keep on saying. So we're gonna put a primer here. Right? Primer over there. So I want you to think this through again. When DNA polymerase 3 continues to build on the primer, is it going to go to the left or is it going to go to the right? Okay, think this through. Which way is it going to go? Okay, don't just look at the previous diagram. Think about it in terms of numbers. Yeah, sure,
Can
go
you
ahead.
repeat the question, please?
The question, yeah. So the question is, okay, everybody understand that this is the primer, right? That's the few nucleotides that's laid down by the primase. Okay. And then the DNA polymerase 3 is going to continue the work. It's going to continue to put nucleotides. So is it going to go this way to the left, or is it going to go to the right? And I just want to emphasize, like double emphasize that when I say the limitation where DNA can only be built in the 5 to 3 direction, I am talking about on the new strand, okay? On the new strand. Okay, I'm gonna like triple circle this guy so you don't get confused. On the new strand. All right, I'm seeing a lot of left. Thank you. Thank you. That makes me feel better, all right? It is true. It's going to go to the left, okay? Because look, if this is 3 here on the top, then this must be 5, right? And then this is going to be 3. So it's going to go 5 to 3 into the fork. Yeah, part of the reason is because it's anti-parallel, but the biggest problem here is that the enzyme, the polymerase 3, is only capable of moving 5 to 3. If the enzyme can go back and forth, then we don't have this problem. Okay, nobody knows why it behaves that way, but it just does. Okay, so that's something that is a bit of a mystery. Yeah, but it's going to go this way. All right. So as the helicase unzip more of the DNA, then it's just going to continue to go into it, right? So this strand here has a name. It is called the leading strand. Leading strand. Okay. It's built continuously. Okay. Towards the replication fork. Okay? It goes into the fork. Remember, that's called a fork, right? So it's going to go into it. It's called the leading strand. Now, at the bottom, similar situation, we're going to need a primer. If that's an RNA primer, I'm not going to label it again. As long as you're using color, it should be okay. Now, I hope you know this time it is going to go to the right, right? I'm not going to ask you to vote again, but it's going to go to the right. Because if you look at the numbers, if it's 3 over here at the bottom, then this must be 5. And then this must be 3. So it's going from 5 to 3 on the new strand. Okay, so what about the rest of these DNA? I don't have the ACTGs written down, but you know it's there. So how are you going to copy all these things as you expose more and more of the DNA? Well, you're going to need another primer and another primer, right? And so on and so forth. And so you're gonna build it not all in one go, but like a little bit at a time. And this is called the lagging strand. Lagging strand. Okay? So it's built discontinuously. Is that even a word? Discontinuously away from the fork. Okay. And the lagging strand contains Okazaki fragment. Okay, so that's an Okazaki fragment, Okazaki fragment. Now, if you follow along, you can see the left side here is the lagging strand. However, on the other side of the bubble, this continues to grow into the fork, and actually this piece is being built continuously, isn't it? So on the other side of the bubble, this is actually the leading strand. Whereas now, as you expose more and more nucleotides on the right side of the bubble, you're going to need to have another primer and another primer. And then, of course, DNA polymerase 3 is going to build the rest. And so on the right side here, this is the lagging strand. Okay, and just to reiterate, these are Okazaki fragments. All right, I'm going to pause for a second. There are 2 more things I need to tell you about this thing before the note ends. Just let you catch up and ask questions. This thing is so much harder to teach online than it is in person. Tell me, like, it's just very hard to get it across. We good? right, if we're good, thank you, appreciate that. If we're good, we're gonna zoom in over here. We're gonna zoom in here. Oh, thank you. That's very nice. So we're gonna zoom in here, and I'm just gonna make up a few nucleotides for you. Okay, I'm gonna make up a few nucleotides. We're gonna say 3' to 5', and I'm totally just making these up. Okay, and you can copy them. You should copy them. There we go. And we know there is gonna be a few nucleotides here, right? We're gonna have the primer, for example. Oops. We're gonna have U, G, G, C. Okay, so that's like this pink thing right here. And then over here, that's gonna be like another primer, U, A, C. And for the record, this is 5', 3', 5', 3'. So that's the RNA primer, laid down by the primase, right? The enzyme is primase. Okay, so this part is easy to sort of understand, I guess. You're gonna have the DNA polymerase 3. Don't draw this yet. Just maybe copy whatever's on the screen first with the colors and everything, and then pause and let me explain. This next part is really tricky to explain. Um, Again, virtually, it's really tricky to explain. All right, so here, this darker blue triangle, it's DNA polymerase. Don't draw this, okay? Just watch. And then you decide what to do afterwards, okay? Now, there is another DNA polymerase, which I have not told you about yet. This is DNA polymerase 1. Okay? Let me explain first, and then I will write down the notes for you. So, what you already knew from what I tell you is this, that the DNA polymerase 3 is going to come over here. The RNA primase, the primer is already in place. So the DNA polymerase 3 is just going to continue to basically like build the rest of it, right? So it's going to travel along the uh, original DNA using it as a template, and then it's gonna build the complementary strand. C, T, because it's building DNA, G, A, C. And then it's just basically gonna keep going, right? That's old news. We already know that. Right? Let me just draw another DNA polymerase 3 here. One more time. All right. So what about over here? Well, it's the same story. Okay? The DNA polymerase 3 is gonna continue on from the RNA primer, but pay attention. This is where things get a bit weird. It's gonna keep on going, and then eventually it's gonna hit the primer, right? Ahead of it, so to speak. So it's gonna put some nucleotide T, G, G, C, A, and then it's gonna hit the RNA primer that's laid down beforehand. So what happens when the DNA polymerase 3 gets to a primer, it actually will fall off. And then it will signal DNA polymerase 1 to come along. Okay? So DNA polymerase 1 is going to come over here and it's going to take over. And the job of DNA polymerase 1 is to remove the RNA primer and replace it with DNA nucleotide. Because after all, the primer is just RNA, right? There's uracil in it. You're not supposed to have uracil in DNA. So the DNA polymerase 1 will remove those nucleotides and replace it with actual DNA. Okay? So that's the extra tricky part, I guess. Let me try to write it out for you. As as DNA polymerase, 3 approaches the RNA primer DNA polymerase 3 is replaced by DNA polymerase 1. Okay, just gonna erase these things you don't really need it. DNA polymerase 1, which replaces the RNA primer with DNA nucleotides. Okay. Yes, please copy it. Sorry, I didn't even see your message until now. So go ahead, write down that. Now I'm gonna add an extra note here. Both DNA polymerase 3 and DNA polymerase 1 have spell checking. That's actually what it's called. And autocorrect functions. So it's a lot like typing. When you're typing something and you made a mistake, what do? You press backspace, right? And then you type in the correct one. So when DNA polymerase 3 moves along, putting the C and the G and the A, if it realizes it made a mistake, it's gonna take out the mistake and then replace it with the correct nucleotide. Okay, same thing for number 1. Usually the autocorrect works pretty well, but sometimes they do miss some mistakes and it becomes permanent. And that causes problems, which we will discuss in lecture 7. All right, finally, okay, lastly, I am running out of colors here, but one more enzyme. Lastly, DNA ligase, which is an enzyme that ligate is to connect. So DNA ligase connects the Okazaki fragments together. Yeah, and the process is done. Understanding this process, like doing research to figure it out, took like decades of work. But ultimately, that's what happens in prokaryotes. Now, there's one final thing I want to mention before we go back to the PowerPoint slides. I keep on saying that this happens in prokaryotes. Do you remember the structure of prokaryotic DNA? Um, actually, it's kind of a big question. I'll just tell you again. Like, it's circular, right? Right.
Circular.
Yeah, thank you. Okay, so I'm just gonna draw it here. Okay, and it's double-stranded. So like technically, I'm supposed to draw another loop, but I want to keep it simple. Okay, so like just circular DNA. In prokaryotes. And so this is kind of what happens, okay? Just watch first. You're gonna have a primer, okay, and then you might have multiple primers, okay? Never mind the 5' and the 3' um for the moment. That's not the point. The point is this is gonna keep going, keep going, keep going, keep going, and then number three is gonna fall off. Number one's gonna take away the primer and change it to DNA. Ligase is gonna glue it up. And then here is gonna go, 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 go. Number three is gonna fall off, tell number one to come over, and so on and so forth. And it's wonderful for the DNA to be circular because you just kind of loop back to your starting point and basically complete the whole circle. All right. Keep this in mind when we talk about humans or like animal cells in general. Our DNA are not circular. Ours are linear. And so we are not able to loop back like the circular DNA. And that actually creates a big problem for us. And we're going to look at the big problem probably in a bit, okay? So that's that. Now I'm gonna post this note after class, of course, but I'm just gonna post it with all the other stuff that we did last class as well, okay? So when you open up the notes, it might look like it's the same note as last time, but just scroll down, okay? And then you will see the rest of the note. All right, let's go back to the PowerPoint for like a couple minutes before we continue. All right. So here is like a professional drawing, if you would. I always like to say it like that. They actually made a mistake in this diagram, and I never remember to correct it. If you look at the purple strand, which is the original DNA, this is 3' here. So just follow it along. You don't draw it on yours. You can just watch me. You can keep on following it, da, 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 da. And then actually it did a little twist here. Not sure why they decided to do that. But then after they do the twist, they forget to change the number. Right? Because if it's 3' here, this is actually 5'. Okay, which means this is going to be 3'. Okay? Not like a super big deal. Like, just want to change it in case some of you figured it out and thought it was weird. Okay, let's see if you can identify some of these things, eh? Like, what do you think number two is? That little orange oval thing? What enzyme do you think that is?
Helicase.
That is absolutely correct. That is helicase. Okay, what about number one here? Anyone else want to try what number one is? That's
Gyrase.
the one. Yep, exactly. Gyrase. And these little orange balls, number three, what are they?
Single strand binding protein.
Exactly, right? Which keeps the two strands from coming back together, right? And then the rest of them is labeled for you already, okay? You have the ligase here. Don't worry about the 5, 6, 7, stuff like that. Those are actually just like the 1, 2, 3 is actually not asking you to label the proteins, is actually describing what's happening in the replication process. But yeah, don't worry about it. Like I'm not going to ask you to label something like this on the quiz. And you can try to look at some of the other stuff though. Okay. All right. So that's prokaryotes. And it turns out what happens in eukaryotes, such as human cells, are very similar to the prokaryotes. Right? But I'm going to tell you three key differences. Number one, eukaryotes, that's us, okay? We have linear chromosome, and we actually have a lot more base pairs to copy compared to the bacteria. Right? Last class I told you the E. coli entire DNA has about 40 million base pairs, but for us we're talking about like 3 billions. So it makes sense for us to have multiple origin of replication. Okay, so think about it. If it's like super, super, super long, and then you have just like a single bubble here, don't write this. You don't have to draw this, okay? If you just have a single bubble, and then you wait for it to like expand, it's gonna take forever. So for us, let's have like more bubbles, more bubbles. Let's create multiple bubbles, and each bubble will expand, and we can copy multiple sections simultaneously. Okay? It's like if I ask the whole class, to come together and then copy the textbook, right? It would make sense for us to each start at a different chapter and then put it back together, right? This is kind of like that. You're going to create the origin of replication at multiple locations, right? So there will be some enzyme copying this part, some enzyme copying this part and this part. And as the bubble expands laterally, horizontally, they will eventually merge together, and then you get the two daughter strips. Okay? Eukaryotes, bacteria, they don't do this. They only have one replication bubble, and it's okay because they have smaller set of DNA to copy, right? So that's the first difference. We have multiple origin of replications, sometimes as many as 100,000 of them at the same time. Second difference, most of the enzymes in prokaryotes have a eukaryote counterpart, but sometimes the same enzyme is represented by multiple enzymes. For example, in prokaryotes, there are only two DNA polymerase, 3 and 1. For humans, we have 5 different DNA polymerase. They all do similar things. We don't have to know what they do specifically. They make DNA basically. Right? So there are 5 versions of the same enzyme. And it's, understandable. Once again, eukaryotes are more complicated. There are more DNA to copy, so it's not surprising to have more complicated tools, right? Complicated enzymes to achieve the same outcome. And the last difference that we are going to talk about is the most important difference. We're actually going to be spending, I don't know, 15 minutes talking about it. It turns out every time we copy our DNA, we are going to lose a bit of DNA each time. Right? So we have linear DNA like this, and you copy the DNA, it gets shorter, it gets shorter, it gets shorter. Every time you divide your cells, every time you copy the DNA, it gets shorter. Why? Right? To understand why, we're going to have to write more notes. Right? So let's go back to our drawing board, and we're gonna draw some eukaryotic DNA. Okay. Which is linear, right? E.g. human cells. Now it's gonna sound like I'm repeating myself, but just stay with me. It will make sense in the end. I'm hoping it will make sense. Okay, so just for a change, I'm gonna do the number a little bit differently this time and see if you can follow. Okay, let's say I put 3 prime here and 5 prime here. I'm sure you understand over here is supposed to be 5, right? And 3. That part, I am very optimistic all of you understand. So we separate the strand, we got the bubbles and whatnot. And now I'm gonna just randomly put a primer here. And then the $1,000,000 questions that I want you to tell me is it going to go that way or is it going to go that way? Let's do a poll and see if it gets better this time. Okay. The pink is the primer. Polymerase 3 is going to come and continue to build upon the primer. Which way is it going to go? Okay, I have about 8 answers. We have 18 people here, so please choose something, or you can message me instead if you don't see the poll. But I need to know what you think. Okay, I'm gonna wait a few more seconds before closing the poll. Okay, unfortunately, it's half half. Half of you say it's A, half of you say it's B. Okay, so maybe it's not still entirely clear, right? Let's take a closer look at this. Okay, don't just memorize the top is always going to the left or something like that, right? Like because the numbers can change, right? And I actually did change the number this time. So let's take a closer look here. Okay, when the RNA primer when the RNA primer is created, okay, so like it's a few red nucleotides, so to speak. Um, in terms of the numbers, what number should I put here? 5 or 3?
3.
3. Okay. And then over here, it is 5. Right? It's always opposite to the top, right? Okay, so if it's 3, 5 like this, then the bottom is 5, 3. And then now we are gonna build a strand. Which way is it going to go? Well, new strand can only go from 5 to 3 on the new one. So it's actually gonna go to the right. All right, so you're gonna need another primer, and then you're gonna need another primer, and then it's gonna all go this way and this way. Okay, I'm gonna put more numbers for you to see. You might have to draw this out on your own later on for it to make sense. Okay. Now let's look at the bottom and think about it. Okay, you're gonna have multiple primers because you don't open up the bubble all at the same time, right? So you need to have multiple primers. And this time, I hope you understand it is going to be going to the left. Okay? I think the biggest confusion for a lot of people is you are reading the black numbers on the original and thinking it's going from 5 to 3. But I keep on emphasizing we are talking about 5 to 3 on the new strand, on the one that you are building. Okay? It's very confusing. I know, I know. If you think of a better way to phrase it, please help the class out. Okay? So what is the problem? Here's the problem. Over here, focus on this part for a second. The DNA polymerase 3 will fall off and number 1 will come and you will replace this primer with actual DNA. You understood that. Same thing here. As the DNA polymerase 3 comes and approaches the primer, it falls off, and number one's going to get on and replace the primer. However, at the very end over there, this RNA primer will not be replaced with DNA because no polymerase is coming, right? This is the end of the line. It's not like the circular DNA where it loops back on itself and eventually you end up where you started. In this case, when it's linear, at the end, it is the end. There's no polymerase 3 coming from upstream, the term is, okay? There's no polymerase coming upstream. And if polymerase 3 is not coming, there's no signal for polymerase 1 to get on and replace the primer. And so that primer is not replaced with DNA, and eventually the primer is just going to be gone, and you lose that chunk of DNA at the end. Same situation on the other side, okay? Will be lost. And that is why linear DNA has a problem that circular DNAs don't. Okay. Every time we copy our DNA, and that happens every time you want to divide a cell, you need to copy the DNA, right? The DNA gets shorter and shorter and shorter and shorter. Okay. And you can understand why that would be a problem. Think about a textbook, right? Let's say every time you read your textbook, you have to rip out a couple of pages from the beginning and from the end. Okay? So it's gonna be okay if you do it for like a couple times, because no one ever reads the, glossary or the table of contents and whatnot. So you lose those pages, you don't really care. But if you keep on doing this, eventually you're gonna lose part of chapter one and part of the last chapter, right? Eventually you're gonna lose valuable information. So that's what happens. If we don't do anything to protect the ends of a DNA, then we risk losing important genetic information. And as we all know, DNA codes for the instruction to make proteins. So if you lose the important DNA, we basically will not be able to make some proteins in the body. And that, of course, is going to be a problem. So after the break, we will look at what measures are available in the body to delay this process and perhaps to tweak it so that it's not going to be as much of a problem. All right. Let's just take 5 minutes, okay? We still have like a ton of stuff to talk about. And meanwhile, please spend some time to think about this 3' 5' business. See you all in a bit. All right, let's keep going with the note here. All right, so as I was saying, having linear DNA has a unique challenge in that we are not able to copy the very end, and as such, every time we copy the DNA, we end up losing a little bit of it. Okay, and of course, that's not ideal. So, what nature has provided us with are these things called telomeres. Okay, telomeres. Telomeres are non-coding DNA that exist at the ends of each chromosome. Okay, so we have 23 pairs of chromosome, 46 chromosomes in total. At the ends of each of these chromosomes are non-coding telomeres. They are basically just repeats of 6 nucleotides, TTA, GGG, and it's going to be repeated, a couple hundred times. Okay, TTA, GGG, TTA, GGG, just like that. And they don't do anything other than being there at the ends of the DNA, and basically delaying the erosion of actual important DNA. So this picture shows you all the chromosomes, and there's a technique called like fish, F-I-S-H, fish, that allows you to light up basically any part of the chromosome you want. And in this case, they light up the ends of the chromosome, right? Which would be where the telomeres are. Right? So you can see it's always there at the very end. Think of it as like the plastic thing that's at the end of your shoelace. Right? The plastic thing protects the shoelace so that it's not gonna fray and unravel, right? In the same way, the telomeres are there to protect the ends of DNA. So let's say every time you tie your shoelace, you're gonna have to cut off a little bit of this plastic thing, right? So you get a brand new pair of shoes, brand new plastic thingies, you wear it, you tie your shoelace, you have to cut some off. Next time you wear it, you got to cut some off, cut some off, cut some off. So you can do this a couple of times, but eventually, you're gonna wear it so many times, you're gonna cut off so much of the plastic thing that if you wear it one more time, you are actually going to cut into the actual shoelaces, right? So that's the situation here. It's not that we are stopping the DNA from getting shorter. It is still getting shorter every time we copy it, but the stuff that we lost from the shortening are not important things. Okay? When a cell has telomeres that have become way too short, we say it critically short, then the cell is not allowed to divide again. Right? So after a cell goes through like 30, 40 rounds of division, the telomeres are going to be too short now. That's a signal that would prevent the cell from doing another round of division. Because if we go through another round of division, it means we have to copy the DNA one more time, and we are going to run out of telomeres. We're going to actually lose actual DNA. So that's not allowed. So the cell becomes senescent, we say. It's basically like it becomes old. And as we age, the pool of old cells is going to increase over time. And that partly contributes to the process of aging. So I have a question for you. We're going to talk about the sheep in a second, okay? But I have a question for you. I don't know, the median age of people who take this program is about what, like 20 years old, mid 20s, early 20s. Maybe some of you are a little bit older, maybe even younger. But my point is, if you're around 20 years old, that means your cells has been dividing, the ones that are alive anyways, they have been dividing for like, some time. Okay, so the DNA in those cells are like shorter than when you were born. Make sense? So how is it that when you have a kid, okay, either you already have a kid or later on you're going to have a kid, how is it that they are able to continue this process? Okay, like don't they inherit our DNA, right? So if our cells were this long and then by the time we have kids, they become this short, right? Don't we pass on like the short DNA to the next generation? And then therefore they would have like less cycles left for their cells to divide? Do you understand the problem I'm trying to say? If not, just say no, and I'll try another way. No? Okay. So basically, uh, this is like your DNA. And then as you divide, you see that it gets shorter and shorter and shorter, right? Okay. So let's say you decide to have kids here, right? When you pass this to the next generation, then they would start with shorter DNA, right? And now they have less cycle left to divide before it gets into the actual DNA. To use the shoelace analogy, okay? You've been wearing your shoes for some time now. Every time you tie your shoelace, you cut a bit of plastic off, okay? And then halfway through wearing the shoes, you decide to pass the shoe to someone else. So that person who inherited your shoe, they will not have the brand new plastic thing. They will have the shortened one. So in other words, the lifetime of the shoe is much shorter than what it could have been, right? Because they inherited the short plastic thing. They inherited the short telomeres, right? Is that a little bit better? Follow-up questions? No? Yes? Okay. So obviously, still confused. Maybe you can ask me a question so I can help you out. Okay, I'll get to that question in a bit, Sheila. Great question. Izelny, do you have a question for me? I'm sorry if I'm pronouncing your name wrong. Um, type a question, okay, if you have. Okay, so this actually, even though in theory sounds like a problem, but in reality is not going to be a problem because of this thing called telomerase. Again, I know things are already confusing, and this potentially can make it even more confusing. Do not mix up telomeres with telomerase. Okay. Telomeres are the repeats that are at the ends of the chromosomes, and they get shorter and shorter every time your cell divides. Telomerase is the enzyme that will rebuild the telomeres. Okay. So without telomerase, the telomeres get shorter and shorter, but with telomerase, you maintain the telomeres length. Right? So telomerase turns out it's something that is only produced in certain cells in the body. One such example would be the germ cells. Okay. People get very confused with the word germ cells. They think it has to do with bacteria. It's not. Okay. In the body, we have two kinds of cells, somatic and germ cells. Germ cells are cells that will become sperm in male or egg. Right? These are the cells that will become reproductive cells. Somatic cells is everything else. So like your skin cells, brain cells, heart cells, liver cells, those are somatic cells. And they do not have telomerase. Germ cells have telomerase because you need to reset the telomere length before you pass it on to the next generation. Okay. So to answer your question, Sheila, it's not true because of the telomerase. You basically reset the situation before you pass it onto your offsprings. Okay. So
So back
mhm,
to the analogy with the shoe sock.
shoelace, yes.
Yeah.
So
How to understand?
so it's like this. Okay, I wear my shoes for like 2 months. I've been cutting off the plastic, and now I want to pass the shoe to someone else. But I'm nice. Before I pass it on, I change the shoelaces, okay, so that it has brand new plastic things, and now they can cut the thing on their own from scratch, from beginning, right? Does that help?
Got it.
All right, excellent. Okay, we're going to talk about Dolly for about 5, 10 minutes. What regulates telomerase activity in germ cells? Excellent, excellent question. Okay. Hold on to that thought. We're going to come back to it in after Dolly. Okay. I'll talk about that because we have a slide coming up, actually. So, Dolly, if you're a little bit younger, you might not have heard of Dolly, but anyone at all? Like, know what Dolly is? You might have heard of it. Like a thumbs up if you have. Yeah,
I have. It's a sheep that they clone, right?
that's right. Yeah. So this happened
Okay.
in 1996, which is like almost 20 years ago, right? And this thing, this sheep is the first mammal that was cloned, right? And the process is what's called nuclear transfer. So the dolly, the sheep, only lived for 6 years, which is half of the expected lifespan. Now, this was not because there was some like technical error in the procedure, or like they made some kind of mistake that caused it to become sick or anything like that. It's actually related to what we are learning. And so let's take a look on Dolly. I hope this is gonna make it interesting and relevant, okay? Trying my best here. So Dolly, the cloned sheep. And the method they use is called a nuclear transfer. Hey, just a random question for you to think about. How many sheep do you think it took to clone Dolly? I'm not talking about all the failed experiment where the sheep might have died or something like that. Like in the successful scenario where they actually make Dolly, how many sheep does it take to make Dolly? Just throw me some numbers, okay? Yeah, 2, 1, many, right? Okay, that's good, right? Keep it in your mind and see. So let's start with sheep number 1 here. Okay, draw your best sheep. Okay, so this is sheep, and probably not too happy to be in this experiment. But this is gonna be an egg donor. Egg donor. Okay. Someone asked me one time, they're like, what? Sheep has egg? Right? I'm like, no, 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 not like chicken eggs or like, duck eggs or something like that. Like, like humans have eggs too, right? So I just want to throw it out there. We're talking about like, sperm and egg, right? So this is the egg donor, and they took an egg from the sheep and removed the nucleus. Okay, just a bit of like background. I'm sure you know this already, but to make a baby, you need the egg and you need the sperm. Egg has half the genetic material and the sperm has the other half, and then after they come together, you get the full set, right? So over here, they took the egg and they removed the nucleus. Okay? So we basically end up having just an empty egg, okay? Like the eggshell, if you would, okay? No genetic material. That is our sheep number 1. And it's time to bring in another sheep. So this is the nucleus donor. Nucleus donor. And from the nucleus donor, okay, they took a somatic cell. And remember, somatic cell is just a cell that's not a germ cell. It's like a bodily cell. That's what it means. Okay, like could be from the liver, could be from the skin, could be from the kidneys, what have you. Okay, so they took a somatic cells, which would be something that has a full set of chromosomes, right? All your cells in your body has a full set of chromosomes, except for the sperm or egg. So if you take a somatic cell, it's gonna have a full set of chromosomes. So took the somatic cell and extracted the nucleus, right? Which contains a full set of chromosomes. Obviously, sheep has different number of chromosomes than us, but nevertheless, it's going to have a full set of chromosomes. So this is going to be the nucleus. All right. You can probably guess what happens next. What they did next is they took the nucleus and then they put it into the eggshell. Right? Combine. And if you wonder how they do it, they literally do it under the microscope. They have these little very fine instrument, and then they literally inject it. You probably have seen it on TV at some point, like in some sci-fi movie or something, except it's real. It can be done, right? In vitro
Kind of like,
fertilization.
sir, kind of like IVF.
Yeah,
Is it?
yeah, exactly, right? We said it at the same time, right? IVF, in vitro fertilization, right? That's exactly what happens, okay? So then we have the egg, and then we have the implanted nucleus, right? So in this situation, basically what they were trying to do is they simulate the fertilization process. Okay, no father was involved here. There's no like male sheep involved. Okay, but by replacing the egg nucleus with a nucleus that has a full set of chromosome, they are basically simulating the fertilization process. Okay, so far, uh, I don't think it's too complicated to understand, I hope. Then we're gonna have a third sheep. Okay, what do you think this third sheep is for? And what do we need a third sheep for?
To grow
Hmm.
the egg. Mm.
That's right, to grow the egg, right? So what do we call people, or in this case the sheep, that grows a
But
baby for someone else?
carrier,
Surrogate?
Surrogate,
Surrogate.
yes,
Yeah.
surrogate.
surrogate, yeah.
Yeah. So this is a surrogate sheep. Okay, and then after some gestation period, we have Dolly. There you go. Dolly. Now, some misconception out there, right? Like cloning does not mean you're gonna have like the adult version immediately. Like if you were to clone me or something, not sure why that would be a good thing, but if you were to clone me, you're not gonna have like a grown up me immediately, right? It's gonna be like a baby version of me, which might or might not grow up to look like me even. Because even identical twins, when they grow up, they might not look so identical afterwards, right? Environment has a lot to do with shaping how you look, right? But it's not just the looks we're talking about, right? It's about the genetic makeup. Okay, it's a clone because it's genetically identical to its mother or father in some cases. So my question to you is Dolly a clone of sheep number one, number two, or number three? Let's do a poll and see what you think.
2 and 3.
Okay, if you think it's more than one, feel free to choose more than one.
I think it's 2 and 3 because the
Okay,
nucleus donor, the surrogate, you know.
could be. Let's see what other people think.
So the numbers are, we don't know,
Yeah, oh,
because
one. Well, yeah, A is one, right? I guess. Sorry.
A is
I
one, two is B, right?
yeah, I should have put
Okay.
A, B, C. Sorry. It's okay, Nestor. I wouldn't know who chose what, but thank you for clarifying. Okay, so a lot of people chose two, some people chose three, some people chose one. The correct answer is actually number two and only number two, because number two is the only sheep that is providing the genetic material, right? Okay, the surrogate, yes, carry the baby to term, but in terms of genetic contribution, it didn't, right? So Dolly is a clone of number two, the nucleus donor. Okay? All of his genetic material is actually from that cell that we harness the nucleus from. Okay? Right, same
Sir,
thing. Yeah, sorry, go ahead.
how long did it take to clone Dolly?
Oh, like the you mean from start to finish, like
Yeah.
to grow a sheep? Yeah, the actual implantation is very quick. Okay, once you figure out how to do it, right? And I don't actually know off the top of my head how long does it take for the baby sheep to grow to full term, but it would be the same amount of time. Okay, but of course the actual research to get it to be successful and so on and so forth, that I would imagine takes like decade of work. Okay, so like just like a real person, right? Like if some people cannot carry their own baby and they hire a surrogate to do it, the surrogate mother has no um genetic connection to the baby, right? Okay, it's the same idea here. All right, back to what we are talking about, right? So Dolly only lived half as long as a regular sheep would, okay, 6 years, instead of like 12 or something like that. And why is that the case? Well, the nucleus that was used to create Dolly is a nucleus that's taken from a somatic cell from an adult sheep. So that adult sheep, the cells in that sheep had been dividing for some time. And so the telomeres in the chromosomes are shorter compared to a newborn. Okay? And so when we take the nucleus from that cell and we grow the dolly from it, genetically, it has the same old age as the adult sheep. Even though it's born like as a newborn baby, but the telomeres are still short. It's the same length as the adult sheep because dolly was not created with sperm and egg, which means they did not reset the telomeres, right? It's still shortened. So the cellular age, right, of the sheep is that of an adult. So it's not surprising it only lived 6 more years, as opposed to, the full life. Does that make any sense at all? Right. So is like back to the shoe analogy. It's like now passing on the shoe without replacing the shoelaces, right? So it doesn't have as much life to go as it otherwise would have. Okay. Any questions about Dolly? Why can't you extract the nucleus? Yeah, I'm sure they could have, right? Like that someone asked in the private message, why can't they just do it from like another baby sheep, right? They absolutely could. But I think they were just like focusing on like the cloning aspect of it. They weren't exactly trying to create a sheep that would live, they just wanted to know whether they could clone or not, right? Basically. Okay. That's that. Now, here's another thing. Some people argue, well, not argue, but like some people have this view, okay, and it's about telomerase. Um, so we understand up to this point that short telomeres has to do with aging, right? And we also understand telomerase is gonna able to reset the telomeres in germ cells and pass it on. So the logic goes like this. Wouldn't it make sense for us to try to use telomerase in like regular cells so that number one, maybe we delay the aging process because we can keep the telomeres long and the cells can, stay young for longer periods of time. Or number two, maybe we can de-age a little bit, okay? Undo some of the effects of aging by providing telomerase. Because if the cells have reached critically short telomeres, then maybe we can reset it and the cells will be able to go on for a little longer, right? That's the logic, right? And it's a sound logic. But it turns out that is not a good idea because telomerase, when it's turned on, when it's produced in a cell that's not supposed to have it, it turned that cell's cancerous. Okay? Now, let me just step back a little bit and give you a better explanation. Someone asked earlier, what regulates the telomerase activity in germ cells, right? Okay, so you have to understand every single one of your cells has the exact same DNA. Okay, whether it's brain cells, whether it's like, kidney cells, liver cells, lung cells, they all have the same set of DNA. And DNA codes for the instruction to make proteins. Okay? So think of it as a recipe book, and it has all these recipes to make all these different dishes. Okay? So every single cell is going to have the same recipe book. But here is the thing. The recipes that you make in a brain cell is not necessarily the same recipe that you're going to be making in your liver cells because the needs are different, right? So the proteins you need in the brain are different from the proteins you need in the liver. And so even though you have the same DNA, which part of the DNA you turn on or turn off, that's going to be dependent on the kind of cells you're looking at. Okay? Which recipe you're going to make is going to depend on which cell you're talking about. So even though all the cells have the DNA to make the telomerase, not all of them do. Okay? The germ cells will be able to make it because they need it there. They need to re-lengthen the telomeres for the next generation. So it makes sense to turn them on there. But if we turn on telomerase in another cell, then that cell becomes a cancer. It could potentially become cancerous because now there's nothing that would stop it from dividing. Okay, so normally we have a cell and it would divide, and then that would divide again. Okay, and after many divisions, right, the telomeres would be critically short, and then it would stop dividing. But if telomerase were active, then the stop is not there. Then they would just keep on dividing, dividing. And when cells have this uncontrolled cell division, you actually end up having a tumor. Okay? Tumor is not necessarily cancerous, but it could be. Okay? We're going to talk more about cancer later on towards the end. Right? So this is true. When they look at cancer cells, a lot of the cancer cells have telomerase in it, and you're not supposed to have telomerase in regular cells. You're only supposed to have them in germ cells, right? And actually stem cells as well. We'll talk about stem cells later on. So a lot of researchers are trying to explore telomerase inhibition as a potential treatment for cancer. Okay, if the telomerase is what's causing the tumor cells to be immortal, to be able to divide again and again and again, then maybe it makes sense to block them, right? Find a drug that would inhibit them. Of course, the side effect would be you could potentially cause the person to become sterile, right? Not able to have babies. Right? So that's the relationship between telomeres, and cancer. Okay, so we have some questions here. I'm just going to write down the answer. You can do them later. Let me see. The answer is B. Just write it down somewhere and then check it later. B and B again. And this is D. Okay, so the three questions B, B, D. Okay, just write down somewhere safe, and then you can do the questions later and check it. One more question. A. Okay. Just kind of running low on time, so I want to skip these things. All right, so the last 15 minutes, I get 10, 15 minutes, we're going to talk about the cell cycle and mitosis. Right, just very briefly, in the cell theory, we know that all living cells come from division of pre-existing cells. Okay, we don't know where the first cell came from. I mean, some people have some theories, but we know all the new cells has to come from some other cell that was already there. And the purpose of cell division is to replace lost or damaged cells. We need cells to grow, help us grow, help us develop. We have a lot more cells now than when we were babies. And for some organism, they use cell division as a way for reproduction. Okay, so that's not necessarily true for all organisms. Like for humans, we don't use cell division for reproduction. We do sexual reproduction, right? Where you need two individuals, each contributing half of the genetic material. But for some organisms, like these ones, they do asexual reproduction. There's no sperm egg involved, and they could do it all by themselves. Okay. Did you know if you cut a sea star, starfish, in half, each half will be able to grow back the missing half. Right. So you end up have two sea stars. Right. So that's an example of reproduction, actually, asexual reproduction. It's the different. It's not the same from as a earthworm, right? If you cut earthworm, the head will grow a tail, but the tail will not grow back the head, right? So that's like regeneration. It's a little bit different, okay? But starfish can reproduce uh, asexually, okay? Plants can reproduce asexually. If you have any plants at home, you know you can just break off a bit of it and then give it to your friends, right? And then they could grow a whole plant from it. Right? Like I have a lot of these aloe vera plants, and then it's just kind of, I keep on giving it to people, but it just keeps on growing and growing, right? So it's a great way to pass it along, right? It's fast and it requires less energy. If you think of like a bacteria or something like that, it's able to copy itself under ideal condition, like E. coli, for example, can copy itself every 20 minutes. Okay? That's really quick, right? For humans, if we want to have a baby, it's gonna take like 10 months, right? Okay, and then you gotta raise the baby. It's a lot of energy, a lot of resources to do that. So that's one advantage for doing asexual reproduction. The biggest disadvantage is low genetic diversity. Okay, you're not gonna be able to create too much variation. So if there is one like a bad mutation in the cell, all the cells that comes after that is gonna carry the same bad gene. Okay, but we will see later on in the course rapid division is actually a good way. It can be a way to introduce genetic diversity. Okay, so this last point here is a bit of a paradox. Okay, and I'm gonna explain more when we get to the right place. Right, but for now, just understand the purpose of cell division. Replace loss or damaged cells. So if you cut your finger, you need new cells. You're gonna do mitosis, growth and development, and for some organism, reproduction. This picture just refresh your memory on what DNA looks like. Of course, it's a double helix, but we cannot fit all the DNA into the nucleus without first packaging it, right? And if you remember, in the nucleus, they exist as chromatin, which is basically just DNA wrapped around these histone proteins. At the time I told you it's like a yo-yo, right? You have the yo-yo thing, and then you have the rope, and then you can wrap the rope around the yo-yo to make it nice and neat, right? And that's basically what the chromatin is. And when you need to clone the chromosome, copy them or like move them during cell division, then you're gonna condense it even further. So chromosome and chromatin are essentially the same thing. It's just that one is much more compact than the other. The chromosome being the one that's more compact. Okay, so this is an old slide. We talked about this already. In humans, you should know by now, we have 46 chromosomes, 23 pairs in total. 23 from mom, 23 from dad, and then when the sperm fertilizes the egg, you get the full set of chromosomes. Of the 23 pairs of chromosome, 22 pairs of them are autosomal chromosomes, okay, or simply just autosomes. They determine various traits in the body. Okay, so for example, your skin color, eye color, your blood type, it determines the kind of hairline you have, right? Like some people have like straight hairline, some people have like widow peak, right? It determines the kind of earlobes you have. Some people have attached earlobes like this. Some people have like ear that has a little lobe there. It's called a detached earlobe, right? So all these external features, as well as internal features, all these things, everything that's unique about us is determined by these 22 autosomes. Okay? So like how tall you are, right? Uh, uh, um, what kind of hair you have, straight hair, curly hair, so on and so forth. All these things are determined by the 22 pairs of autosomes. Okay? The remaining pair, the 23rd pair, is going to be the sex chromosome. Okay? So for female, it is XX. For male, it is XY. So I want to ask you, is it the dad or the mom that determines the gender of the baby?
That.
Is it the dad?
Yeah, the dad, right? Because the dad is the one who has the XY. Okay? Half of the man's sperm is going to carry the X, and if that sperm fertilizes an egg, then it's going to be a daughter. It's going to be an XX. The other 50% of the sperm is going to be a Y, and if the sperm carrying the Y fertilize an egg, then it's going to be a son, right? My grandmother is very old fashioned. Okay, she's like in her 90s now, and I have two daughters. You probably know that by now. And every now and then she would sit next to me at like family gatherings, and she would whisper into my ears like, "What? Why is your wife not giving you any sons?" And then I would have to tell her it doesn't work like that, right? It's actually me. It's actually my fault, right? That we're not having any sons, right? It's the man that determines it. But she doesn't listen. She's like, ah, no, I know. It's not that. I have many sons, right? She would say. So that's one situation where you should know it is the male that determines it. And when I say it's the male that determines it, I don't mean it's not like they have a choice, okay? Like it's all random, but genetically, it's the chromosome that the sperm carries that determines the gender of the baby. Okay. So we're just on our last slide, and then we're gonna be done. It turns out the number of chromosomes is not related to the size of the organism. Okay, some organism is really big, and yet it has very little chromosome. Okay, something that's small like a goldfish, right, has more. Right? So there's no correlation between the size of the organism and the number of chromosome it has. It turns out a lot of these chromosomes, in some cases, they are like duplicates. Okay, they don't actually, they're like fillers almost that are just there. It's think of like a magazine. It has a lot of advertisement in it. They don't really do anything in terms of content. They're just there, right? So that might be one of the reason why something small can have a lot of like filler DNA. Okay. I think that's a good spot to stop. We have a lot of things to cover for the rest of the lecture, but unfortunately we run out of time. So I will see you all next Monday. Please take some time to review the notes. It's a complicated concept, um, and uh, and if you could, please also print out lecture 7. We might start on it. And lecture 7, you really need to have the study guide and the lecture notes, like a physical copy of it with you, because we're going to be solving some problems, okay? And you need to write down these things. Okay, thanks so much. Have a good one, guys. Bye-bye.
Thanks, sir.